So in the last video, we talked about all the different cell types that we are made of. But what is a cell? What is it made of? And how does it work? Well, let's find out. This is the nucleus. The nucleus contains all of our DNA, or genetic code. Now, the DNA is tightly packed inside the nucleus, but if you would unwrap it, every cell contains roughly 2 meters of DNA. Now, we have 40 trillion cells, so you can imagine how many meters are packed inside of you right now. Now, this genetic code is used to make proteins. But how that process works, I will quickly explain before we move on to the next organelle. So our DNA is our genetic code, a code to create proteins, which you can sort of view like the instructions to create a Lego kit, for example. Now, every cell contains the DNA code or the instructions to create all proteins that we as humans can make. However, a cell doesn't need to create all proteins. Actually, every cell produces a different set of proteins. Therefore, we first generate a copy of the code or the instructions for the specific protein we want to create, which we call RNA. And this RNA is then translated by ribosomes, which are proteins that can read the instruction and build up the protein in the correct order. Now, to see where this happens, we go to the next organelle. RNA is translated inside the nucleus and then goes out, where it encounters our first organelle. So this is the next organelle, the endoplasmatic reticulum, or ER. And the ER is covered in ribosomes, which means that the RNA that comes out of the nucleus is translated by these ribosomes, and the proteins are immediately produced inside of this organelle. Now, in the ER, those long strands of proteins are folded with the help of many chaperone or helping proteins, and some additional modifications are added to make them reach their final efficient form. After the ER, some proteins are released into the cytoplasm, but some of them also enter our next organelle. So proteins that are made need to go to different places. They need to go inside the cell or to the membrane, or maybe even be excreted outside of the cell. So this happens in the Golgi system, where the proteins are tagged with specific tags and separated in different vesicles that will determine their final destination. So that's the full cycle of proteins being newly made and transported to their right location. However, as it is important to make new proteins, it's equally important to destroy or get rid of them. And that's done in these. So these small vesicles are our lysosomes. They are like the trash bins of our cell. So they have a very low pH, meaning that the proteins that enter them are degraded or broken down into smaller pieces. And these pieces can be used to make new proteins again. So the cell is actually very efficient at recycling our proteins. Now, to do all these processes, we need a lot of energy. They don't all happen by themselves. And that energy is provided by the powerhouse of the cell, our mitochondria. So our mitochondria create energy, and they do this by charging small molecules, sort of like rechargeable batteries. And these batteries can then, for example, bind to a ribosome, as we talked about, release its charge, and as such the ribosome can perform its function. So the charging of these molecules is a chemical process that is based on the transferring of oxygen. And this is why oxygen is such a vital part of almost all life that we know. So these are the basic components that a cell needs to function. However, if a cell would be like this, it would immediately slump down. So we need some structural components. We have a couple of them, but I'll talk about the two. The first is the actin network. So actin are long strands. And even though I only show a couple of them here, they pass throughout our entire cell. And these long strands provide structural integrity so our cell doesn't immediately collapse on itself. And also some organelles are actually tethered or bound to kept in place by this actin network. And the final thing I will add to the cell are these. If I can add them somewhere. It's getting full here. These white strands are microtubules. And again, these microtubules expand from our nucleus throughout the entire cell. And these are like the highways of our cells. So proteins and vesicles get transported alongside of them to reach the destination. So unlike these static cardboard pieces, these microtubules actually are constantly expanding, breaking down to reach all the areas of the cell where they are needed. And I think that's where I want to end as well, because unlike our textbook figures or this very static piece of cardboard, our cells are actually a highly dynamic environment, constantly changing, constantly making proteins, constantly breaking them down, constantly recycling them, transporting all the proteins, taking things up, excreting things, move about a lot, change shape, but it's not just happening in this cell, but roughly 40 trillion times at a time. And it's crazy to think that it all just works. In most cases. And with that thought, I will leave. Take care of yourself, take care of each other, and have a wonderful day.